morning, children. Um, my name is Sudharani. I would like to explain about the subject biology. So, first we have to 10th class children listen carefully. So, we have 10 chapters in 10th class. So, what are those? Nutrition is the first chapter, then respiration, circulation, and then uh, excretory system, and then coordination, the linking system, and the sixth chapter is reproduction, and the seventh chapter is coordination in the life processes, and the eighth chapter is heredity. In the sense, we have to how the generations will continue, and the ninth chapter is our environment or our concern, and then the last chapter that is natural resources. These are the 10 chapters we have to see and we have to learn from this 10th class biology. And then along with this textbook part, and we have to learn about 29 environmental topics, environmental science. Okay, so first chapter is nutrition. I'd like to explain about the first chapter, that is nutrition. So the food supplying system. So for example, how to get energy. So for lifting or for walking, for running, whatever it may be, we need energy. How we are getting energy. So when we have to consume food, then only we will get energy. So now I'd like to explain about the nutrition. So in our nutrition, what is meant by nutrition? Nutrition in the sense, the substance which contain different type of nutrients. What are those? 7 nutrients are available in our diet. So night is a diet. So what are those? Carbohydrates, proteins, fats, then vitamins, minerals, fibers, and then water. These are the 7 important nutrients in our diet. So they will provide energy to do any works. So here you have to see, are you able to prepare food ourselves? No. Yes, you are right. Human beings, we cannot prepare our food ourselves. So we can depend. So how will we get food? We will get food from plants. So is it true or not? So the green color plants only have the ability to prepare food. So we have to know that. So how the plants plays a major role to synthesize food. So generally smaller organisms to higher organisms, even bacteria, amoeba, and different types of microorganisms also need food for survival. So what is the use of food? Food is help to maintain our body and repair the damaged cells and then provide body temperature. This is the use of diet. And here you have to see how these plants plays a major role. Here you have to see 2 different types of nutrition. One is autotrophic nutrition and the second one is heterotrophic nutrition. So here autotropism in the sense the organisms have the ability to prepare their own food without depending others. So that type of organisms we can call them, that type of nutrition we call them autotrophic nutrition. So the plants only have the ability to prepare the food. So here you have to see some factors are very essential to prepare food. What are those? Here water, and then carbon dioxide, sunlight, and the green color plants only now. Why the plants are green in color? That is due to the presence of a pigment, that is chlorophyll. So here these 4 factors are very essential for preparing food. And the next one is here one more type, that is heterotrophic nutrition. So heterotrophic nutrition in the sense those organisms which are unable to prepare their own food, they can depend on other organisms. Those type of organisms are called heterotrophs, and the type of nutrition we call them heterotrophic nutrition. Here 3 different types of nutrition are there. What are those? Saprophytic nutrition, halozoic nutrition, and then parasitic nutrition. So the first one, saprophytic nutrition. Saprophytic nutrition in the sense here they can break down the large molecules into simple molecules and then they can get the food from dead and decaying organism matters. That is how to see Dead bodies, animal dead bodies, and plant trunks. All these are the food for some microorganisms. What are those? Fungal species, bread mold, then mushrooms. These are the best example for saprophytic nutrition. And the next type is halozoic nutrition. Halozoic nutrition also the organisms cannot prepare the food, but they have to consume food. Those type of organisms are called holozoic nutrition. Here they have to engulf. Engulf in the sense swallow the food. Intaking the food. So here you have to see four steps you have to see here. Ingestion, digestion, and then assimilation, and then absorption. These are the four different types of steps you have to see in holozoic nutrition. So the best example for this is human beings and then animals. Along with this, you have to see single cellular organism, that is amoeba. And the next one is parasitic nutrition. So parasitic nutrition in the sense these organisms directly depending upon the other organisms. So they have to depend on host who will provide shelter to them. Those type of organisms are called parasites. The type of nutrition we call the parasitic nutrition. So the best here you have to see plants also. Some plants they cannot prepare their food. The best example for this plant is cuscuta. Cuscuta cannot prepare its food. It can depending on host plant only. And then next one is lice, head lice. It can suck our blood. Human beings, animals are the best example. Bugs. All these are the best example for parasites. And then the type of nutrition we call the parasitic nutrition. So now I would like to explain about the first type, that is autotrophic nutrition. In this autotrophic nutrition, plants plays a major role. So we have to call them, plants are the universal food providers. What we call them? Plants are the universal food providers. How can we say like that? So directly or indirectly, most of the human beings, animals, all these are directly or indirectly depending upon these plants only. Okay. So here, before going to that, how can we prove that? So science is an experimental proof. When we have to do this process by using an experiment, then only we will prove that that is a proper result. So here also you have to go through an experiment. So before going to that, what is photosynthesis? What is an equation for photosynthesis? A scientist, C.B. Van Niel. He only derived a simple equation. That simple equation is here CO2 is present in the atmosphere. Yes or no? Air only na? So what is the components of air? Nitrogen, oxygen, carbon dioxide, argon gas, some dust particles. All these are the components of air. So here 0.03% of carbon dioxide is available. So this carbon dioxide helps to prepare the food when it is combined with water. So here CO2 plus 2 molecules of H2O gives rise to. So when the process takes place, when the photosynthesis takes place, so when the sunlight is available, then only sunlight and then water, carbon dioxide, only in green colored plants. So there only the photosynthesis process will be takes place, and then only the plants can prepare their food, and the food can be stored in different forms. So some plants they can store the food in the form of roots, leaves, fruits, vegetables, like that, and underground stems also. So here 1 molecule of carbon dioxide is combined with 2 molecules of water under the presence of sunlight. So here you have to see that carbohydrate, water, and then liberation of oxygen. Oxygen can release in the atmosphere, so that only plants helps to prepare the food and can purify the atmosphere also. So exchange of gases will take place. It leads to purification of the environment. So this is proposed by a scientist, that is C.B. Van Neel. C.B. Van Neel in the year 1931. So, and he only did an experiment in purple sulfur bacteria. Purple sulfur bacteria. So, here instead of water, this purple sulfur bacteria can use H2S, carbon dioxide plus H2S. So here there is no liberation of oxygen, you have to see here. Why? We are using H2S instead of water. So this is an equation for photosynthesis in case of purple sulfur bacteria. And then this is balanced by one more scientist. His name is Robert Hill. Balanced equation is derived by Robert Hill. What is the balanced equation? 6 molecules of carbon dioxide is react with 12 molecules of water under the presence of sunlight only in green plants. That is, chlorophyll is the pigment in green plants. So sunlight. So what are the end products you have to see here? Glucose, C6H12O6 plus 6 molecules of water, and then oxygen is liberated in the atmosphere. This is the equation for photosynthesis. Okay, so how can we prove that the plants are able to prepare their food? So we have to do an activity, experiment. So now I would like to explain about presence of starch in leaves. So in previous class, we already studied about food components. So here we have to see, if we take a potato and then add a few drops of tincture iodine, then the potato is in creamy in color. Then we have to add a brown color tincture iodine, automatically it can turn into blue-black color. So what it indicates? The potato contains carbohydrate. So it indicates potato is a carbohydrate rich food item. Okay, so that one we already studied in previous class, 7th class. So now I'll explain about the first activity, that is confirmation test. If the plants are able to prepare their food by using these 4 factors, yes, they have to prepare. So presence of starch. Starch is a polysaccharide now. So starch in leaves. So in plants, which part is able to participate in photosynthesis process? Leaves only plays a major role for preparing their food. So here, so we have to do this activity in this process only. So now I'll draw the picture. So I have to take, what are the materials? First, before going to that, we have to learn what is the aim. Aim in the sense what we are ready to prove. So that is aim. Aim in the sense our goal. What is our goal? To prepare, to prove that. To prove that presence of starch in leaves. Presence of starch in leaves. So next, for conducting any experiment, we need some requirements. Those are called operators or materials required. Apparatus. What are the apparatus you have to use here? One is beaker. Beaker, test tube, then asbestos gas, asbestos gas, and then tripod stand, then Bunsen burner, then a chemical you have to use here. That is, one is methyl alcohol and then methylated spirit. Methylated spirit, and one more, that is iodine. Iodine is helps to confirm the test. That is iodine. And then brush. Along with this, you have to take water. These are the materials you have to use to do an activity. So now you have to arrange the operators. So this is the tripod stand, and then arrange asbestos goes upon this tripod stand and turn on the flame. Like that you have to draw and put a beaker of water. That beaker also you have to fill with water. So why we are not using this beaker directly on the flame? Glass is very sensitive. Then you have to apply directly upon the flame. So it leads to breakage of the glass. And then we are using chemicals now. It will directly apply to the flame. You have to see the bump of these chemicals. So to avoid the problem, you have to use boiling process. So this beaker is filled with water, and then take a beaker, a test tube also you have to take, and along with this you have to take a thin soft leaf which is able to insert into the test tube. So before putting this test tube into the water bath, have to fill the test tube with methylated spirit. What is that chemical? Methylated spirit. So this full beaker is filled with water, and then the test tube is filled with methylated spirit. Along with this, you have to see the leaf. This is leaf, and this is a methylated spirit. Okay, so here you have to boil the beaker. Then what happened? Here the test tube already filled with the methylated spirit now. So why we are using methylated spirit in this test tube? So already plants are green in color. So it contains chlorophyll. So before going to our test, I have to remove the starch from the leaves. So what is the chemical used to remove the starch that is only the methylated spirit. So after boiling this leaf, then what happened? The leaf is turned into pale white. So this one, that leaf will be removed from the test tube carefully and then put on a petri dish. Petri dish. And then what is the confirmation chemical you have to use here? That is iodine is an indicator. So here you have to add a few drops of iodine. iodine. Then what happened? The pale white color can turn into blue-black color. The leaf, which is in pale white in color, then you have to add an indicator, that is tincture iodine. Actually, tincture iodine color is brown in color. So when you have to add to this pale white leaf, then automatically that leaf is turned into blue-black color. So what it indicates? So the leaves have the ability to prepare the food by using different factors. What are the different factors you have to see for the photosynthesis? One is water, carbon dioxide, sunlight. So you have to see all this process in green color plants only now. So these are the four most important factors plays a major role for preparing food. Okay. So what is the definition for photosynthesis? So formation of complex molecules from a simple molecules by using different types of factors. What are the factors? Carbon dioxide, then sunlight, water. So these are the main important factors helps to prove that. Okay, this is about the confirmation test. So did we prove or not? Yes, we already proved that. What we proved? Plants plays a major role to prepare the food. That is, to place a major role to participate in photosynthesis process. Yes. So we already confirmed it. Now I'll go and explain the importance of factors. Okay. So first factor, that is water. Water is the first factor, plays a major role in photosynthesis process. So in previous class, that is in 7th class, so we already studied in nutrition. Yes, yes or no? Yes. So a scientist, his name is Von Hellman. So he only conducted that, but he don't know this water plays a major role for photosynthesis process. But he conducted an experiment on potted plant. What he conducted there? So he took a potted plant, and that potted plant is covered with a polythene cover, and then he did not apply any water to the plant, and then put some small cones upon this cover. Then during the time of rainy season, what happened? The water will go pass through the polythene cover, and then they can mix with the soil, and the plant will get the nutrients from the soil along with the water. So then what happened? The plants can consume the nutrients and able to prepare the food. But he don't know. But what he said? Just water is helps to increase the mass of a plant. So what he said? Increase the mass of a plant. Mass in the sense weight of the plant. So after 1 year, he observed the plant. So what happened? The size of the plant can be increased, but he did not apply any water to the plant. But the rainwater only helps to grow the plant of that potted plant. Then the plant can grow, but it cannot grow completely and well developed plant. Why? So regularly we are not pouring water. Just rainwater only helps to grow. So that little amount of rainwater only helps to growth of the plant. So here you have to see the little amount, increase the mass of the weight. Okay, mass of the plant. So what he concluded that the water plays a major role to increase the mass of the plant. That is done by Von Hellman. And the next one is, what is the one more factor, that is air is another factor plays a major role for photosynthesis. Air. So here Joseph Priestley. Joseph Priestley. observed that hair plays a major role for photosynthesis process, but he don't know what is the experiment here. But the plants cannot plays a major role for this process, but air helps to exchange the gases. So here, exchange of gases, he observed exchange of gases. So he conducted a series of experiments, and in the year 1770 he conducted this experiment, bell jar experiment. What is that experiment? Bell jar experiment. So here he took one mint plant, then mouse, then burning candle. Burning candle. So here he already observed that in the year 1774 he discovered a gas, that is oxygen. Oxygen is discovered by Joseph Priestley in the year 1774, but he did not coin the name for this gas. That is coined by the scientist, that is Lavoisier, in the year 1775. Lavoisier. Okay, so here bell jar experiment, that is conducted by Joseph Priestley. So first he took a bell jar. Invertedly he kept on a floor, and then inside the bell jar he kept a burning candle. Burning candle. So continuously the candle can burn, but after that, along with this burning candle, he kept a mouse. Mouse. So mouse is a living thing or a non-living thing? It's a living thing. It has the ability to respire. So here this is the candle, burning candle. Along with the burning candle, he kept a rat and mouse. These are the experimental animals. So after a few minutes, he observed that the candle can turn off the flame, and then the mouse can be suffocate in the sense that is in unconscious state. So then he did one more experiment, that is he took one more bell jar, and then inside the bell jar he kept a burning candle, and then mouse, along with the burning candle and the mouse, he put a potted plant, that is mint plant. So any potted plant you have to use here. So green color plants only plays a major role for photosynthesis process. So here, how can we, but he don't know that the plants cannot participate in photosynthesis process, but automatically he kept the potted plant. So then what happened, you have to see here. After few minutes, you have to see the candle can continuously burning, and then the mouse is in live condition. So what did you conclude that? So here in the second step you have to see. So what is the gas held to burn the substances? Oxygen plays a major role to burn the substances. So here candle, when it is able to burn, when oxygen can be provided to that candle, then only it is able to burn continuously. So here rat already kept inside the bell jar. So what happened here? the rat can exhale the carbon dioxide. It can absorb, it can inhale oxygen. That is the process we call the respiration, you know. So here what happened? The oxygen is intake by this mouse and then carbon dioxide is exhaled out. Then here there is no sufficient amount of oxygen in the bell jar. So then what happened? Here the mouse cannot be survived. Even the burning candle also cannot burn continuously. It can turn off the flame. So that is due to the absence of oxygen. While this rat also want to require oxygen. Even the candle also want to require oxygen for burning. So here there is no sufficient amount of oxygen. So how it is possible? So here then he kept a potted plant. Then what happened? So we already studied that in photosynthesis process. So oxygen is liberated out during photosynthesis. So oxygen can be released. Then the potted plant, when it can release the oxygen, automatically the candle is able to burn continuously, and then mouse also survive in condition, live in condition. So he concluded that exchange of gases takes place inside the bell jar. That is done by the scientist, that is Joseph Priestley. This is one more factor. The next one is light. Light in the sense sunlight. Sunlight. So sunlight also an important factor for photosynthesis. This is conducted by a Dutch scientist. His name is John Ingenhoudt. Okay, so how he conducted his experiment? So he took a plant and then kept in a water glass. So here that glass can be kept under the sunlight. Then after a few hours, he observed small bubbles observed from that glass. So why he select the glass? Clearly visible to our eyes. And why he select the water plant, aquatic plant? So when you have to select the aquatic plant, we can clearly, actually gases cannot be see visible to our eyes. Yes or no? So to observe this experiment, so science is an experimental proof now. So how can we prove that? When we have to see that experiment with our eyes, with our naked eyes, then only we are able to conclude that experiment. So here also he did an experiment by using an aquatic plant. So he put an aquatic plant in a jar. Then after that what happened? When he kept under the sunlight, he observed bubbles, air bubbles. So these air bubbles are the oxygen will release through this in the form of air bubbles through this plant. So what did you observe? Here also you have to see that sunlight also plays a major role for photosynthesis process. So one more confirmation test also is there for this experiment. I'll explain now. Okay. So here and the next factor, that is chlorophyll.